Good morning again, and welcome to Lecture 3. And in today's lecture, we're going to focus on bacterial metabolism and growth. Metabolism focuses on how they get energy and use energy and to make molecules. And then the growth part focuses on how they use the energy to actually grow, meaning make new molecules and what are the factors, environmental factors that can affect the growth. Okay, and this is a very, very important topic, and it's very essential. So I'd like to get started. So when we think about metabolism, it's thinking about the molecules getting bigger or small, but in fact, the metabolism can be further classified into two processes: catabolism, and which is for energy generation, and anabolism, which is for energy consumption. Now, last week we had a problem that my hand that I'm using to highlight, it's not showing up on the screen. Can anyone tell me whether you can see my hands moving? Right now, I'm circulating catabolism.
No, Professor.
No.
We can.
Okay, so then I'll have to go to use the pen function. Okay, let's see if it will work. Okay, yeah, so now I'm circulating catabolism, and then the anabolism. Okay, so together they are metabolism. So thank you all for responding. Now, so we're going to have to go into this in detail, but to keep that in mind is very important that you have substrate and you have product. And substrate in this case usually is our larger molecule, and you extract energy. So the way you think about this is, how do you know which molecule can generate energy? One of the things you can actually think about is that something can be burned, like the wood, right, can be burned. And wood is what? It's a polymer of cellulose. Cellulose is a polymer of glucose. And so so in that sense, glucose can also be burned, catabolized to generate energy. Now, in general, majority of the energy, majority of microbes use this process of generating proton gradient and the proton motive force, and that's being used to generate ATP. So ATP is not directly, in most cases, not directly generated. And then the proton gradient or the ATP then can be used to take to drive biosynthesis to make monomers into large macromolecules. All right, so that's the distinction and how three terms are aligned to each other. And so when we think about grow bacteria, and this issue of terminology involved, and so you have the and then you have what's called the bacterial culture. So in terms of bacterial culture, the term here is the culture for microbiologists is really a cell suspension. Okay, so bacterial culture just means a bacterial cell suspension of bacterial cells. Right? You took chemistry, and so you understand the distinction between suspension and solutions, right? Then in terms of getting the bacteria to grow, and that we have to provide nutrients, and so that involved culture media, and so these are the terms that you'll run into. So we have micronutrient, and that's required for microbe to grow. And then in terms of the media, it can be further classified as chemically defined or complex media. Chemically defined example is like glucose, a salt with some glucose, and then that's defined. You know the composition, and then you have complex. Let's say using soup, like meat soup, right? And you wouldn't know what molecules inside. And then you have solid versus liquid, and solid use the agar to actually make a solid petri dish, agar in the petri dish, and then you can grow. Liquid usually is, by define, it's liquid. And then in terms of growing, and it's important that you have to use what's called the aseptic techniques, and your professors in the lab will show you more precisely. So the microbial nutrient then involves like carbon source, nitrogen, macronutrient, and micronutrient. So carbon source usually is the most common is glucose. So that's a very important molecule. Okay, glucose is a universal carbon source for microbes. Okay, majority. You will notice I always say majority. I don't say all, because there are some that you can find exceptions that microbes don't use glucose. I'm sure you can find it. So it's safer to always say, well, glucose is the most common carbon source. And then you have nitrogen source. Microbes usually rely on proteins. Then you have macronutrient. You need phosphate, sulfur, calcium, iron, right? And then you have micronutrient. Microbes need those too. So chemically defined, I talked about that, and solid versus. So let me show you some aseptic technique. The key is to keep the microbes that you're studying inside the media, and preventing the microbes from the outside getting into it. Now the key here you have to understand is that in the air, there's microbes in the air, everywhere, right? Okay. So how do you then prevent the microbes from getting in? One way is to get a Bunsen burner. And that we have been using that for 300 years and has been very effective. And so these are the basics. So basically what's happening is this is a flame, right? And the air is going up and then going down, right? And so think about a fountain. You see, in this particular space, the microbes are here. They're being driven upwards. So there's no microbe in the immediate vicinity of the flame. So, but obviously if you go to here, then it's dangerous, because the bacteria in the air may fall into your tube. And also notice the test tube was held not straight up, okay, always sideways, to reduce the area that can have the air or the dust or microbe fall into it. And so your professor will explain a lot more further in detail, and this is how you transfer liquid microbes to liquid and to the solid media. Okay, and so now we're going to focus on how actually microbe will grow use glucose as an example. As I said earlier, glucose is the most common carbon source. So here I use E. coli, well, a bacteria cell as a, this is our bacteria cell, and then you have glucose that's coming in. Now, the glucose had to first get into the cell. Now, once it gets into the cell, it can go to the from glucose to ethanol, CO2, and ATP. This is one way that glucose can be metabolized. And in this pathway is called the Myrov pathway, and it's also called the glycolysis, commonly known as glycolysis. And the most important intermediate in this process is called pyruvate. And the most important end product, okay, here is ethanol, CO2, and 2 ATPs. Okay, this is very important. Now, we're not a biochemistry class, and I will let your biochemistry professor to discuss the biochemical reactions that can actually generate energy. So in this class, we're focusing more on the living experience of microbes. So we are focusing on what goes in and what comes out. What comes out, obviously, is the energy. That's the whole point. And, well, along with the energy, then you have side product or end product and key intermediates, right? So in this process, in the Myrov or glycolysis pathway, or you can call it fermentation, and this is the end product and the intermediates. Then you need to also understand what are the microbes can actually carry this process. Now this process, if it's only this process, then yeast is the representative that can carry this particular process perfectly, right? That's the reason you use yeast to make wine. You don't use E. coli to make wine. Later on, I'll explain why. Okay, so now, so here is the detailed steps are for the Myrov pathway, or commonly known as glycolysis. And so this is stage 1. So glucose comes in and gets phosphorylated, and this phosphorylation is coupled to the PTS. Remember last week we talked about the import of the glucose. And once it's inside, they put a phosphate on, and that's a common process to activate a product or intermediate. And so now you have, so now we have phosphorylated glucose and fructose, and then you get second phosphorylation. And then following that, this intermediate gets split into two, gets oxidized. Okay, let me go back. And so this process, so this molecule gets split into two 3 carbon molecules.
Professor, we can't see your cursor.
Yeah, I know. This is horrible. So this, you have 2 3 carbon molecules. Then in the process, so the next stage is for this reaction to happen, you have to have the oxidation. So NADA is the one that takes on the electron, and you're going to see this NADA, NADH coming up again and again. So this is a very important molecule. So here you have the NDA, NDA is on this side. Then NADH is on this side. So notice that you have two protons gets carried on, and so when in this direction from here to here is oxidation, from here this direction is reduced. And so this becomes very important. This is called electron carrier. And so now if you focus on the next stage, so now you have two NADH, and then that's for the, so these are the high energy molecule that the 2 ATP is released. And then you go through two intermediates, and then two more ATP is released. And so, so in the entire process, what if you took everything in consideration, what you will see is that you can have two stages of glycolysis from glucose to here, okay? And so now, once two ATP is generated, so the key here is that you see two ATP is used, 1 and 2. And then how many ATP is generated? Well, 2 here and 2 here. So in this process, the net ATP is 2, okay, because you use 2 ATP and you generate 4 ATP. So now you net with 2 ATP. Now, in addition, it is important that electron carrier, the excess electron, actually gets used immediately in the process. So the 2-pyruvate can either get to make lactate or ethanol. Now, it is important that you focus your focus your attention on this number 2. So why do you get 2-pyruvate? How do you keep straight? And then you have 2 ethanol, and then you have 2 CO2, right? So the numbers are very important because the way you think about this is you keep track of the carbon. So glucose is a 6-carbon molecule, right? And this carbon has to go somewhere; it wouldn't disappear, right? And so pyruvate is 3-carbon molecules. So now total is 6. And if you come here, lactate is a 3-carbon molecule. And ethanol, you know, is a 2-carbon molecule. So it's 2 is 4, and then 2 CO2. So together they are 6 carbons. Right? So so this is how you keep track of the process and how many things can be generated. Because if you don't keep track, you may think, okay, 1 glucose, maybe 2 ethanol. Then what happened to the other 4 carbons, right? So it is important to keep track of that. Now, I wouldn't bore you with the detail, but you do in the process need to know that these are the names of the intermediate. These are the names of the intermediate. These are the enzymes. Okay. Now I would keep this slide as slide 16 as almost like a dictionary. Right? If you don't, some of you are really interested in the intermediates, and the biochemistry class may ask you, what are the intermediates? What are the enzymes involved in carry out this process? Because that's important too. But for us, for this class, this will be just like a dictionary sheet. You can always come back and refer to if you need to. Okay, so what we just found is you go from glucose, and you go through glycolysis, and then you generate 2 ATP. Okay. Now, is that the only way the glucose can be used to generate energy? So the question was, do we need to memorize all the this pathway enzymes? No, but no. Remember the exam is open book, okay? And I intend to run that as an open book. That's why I may ask you a question of go back to that slide to look up what the enzymes. But I won't ask you to memorize it. Okay. Now, what I ask you to memorize. Now, this slide is a slide you should memorize. You should dream about it, okay? Because this is an overall picture. And so, yes, there's another way glucose can be used to generate energy. So let's follow that before we go into the detail. So you have pyruvate as an intermediate. As it turns out, pyruvate can be further oxidized into acetyl-CoA. Then the acetyl-CoA can be, yes, to answer your questions, on the exam, you can ask me later, but I either will write it out for you specifically, or I'll tell you which slide to look for. Okay, let me restart. So pyruvate is an intermediate, very, very important intermediate. If there's something that you have to remember, as I said earlier, this is the slide that you do need to remember. Because you're going to refer to this a lot more often, and remembering this slide will guide you to learning and focusing on individual components. Okay, that's why this slide is important. So acetyl-CoA is an intermediate, and then acetyl-CoA can get into the Krebs cycle or citric acid cycle, or Tca cycle. There are different names for it. You need to know all of them. You know they're the same. Go through the Tca cycle. The Tca cycle will take the acetyl-CoA and then burn it, well, metabolize it, or catabolize it into CO2 and generates NADH and FDAH. Notice that I did not put numbers in here, okay? That's for you, that I left out specifically so that you can actually go and calculate. That's a very important thing that you need to add back to this particular slides. Then the CO2 is released, it's air, it's completely oxidized. There's nothing you can no energy can be generated from CO2, right? And then the NADH and FDAH are electron carriers, and then they send the electron to the electron transport chain, and then the electron transport chain allow the electron to flow from one end to the other, and in the process it generates a proton gradient, and then the proton gradient is used to generate ATP. And then altogether you generate 38 ATP. So this is a good time to pause and think about glucose utilization. If you are given a choice of one molecule of glucose, what would you do? Do you want to use glycolysis pathway to generate 2 ATP, or you want to follow this complex pathway but generate 38 ATP. Which one would you choose? Anyone? Oh yes, just go ahead, Ashley.
I would choose the 38 ATP, the citric acid cycle.
Mm-hmm. And that's very smart, right? I would tell you, E. coli will do the same. But why is 2 ATP even a choice?
Maybe because there's a lot of byproduct when you use the citric acid cycle instead. So there's more byproduct of something that you don't need.
Okay. All right. Keep that question in mind. All right. So keep that question in mind. Why isn't, so the 2 and 38 are so different. Why would anyone choose 2 ATPs, right? Sean, did you have a comment?
I was gonna say maybe they picked the 2
Yes.
ATPs because they can't do the citric acid cycle. They're lacking oxygen or
Very
something,
smart.
and they can't do it.
You mentioned something about why can't they do it?
If they're anaerobes, they can't do the citric acid cycle, right?
Aha, very good. We're jumping ahead, but the oxygen is key here. So let's go through the detail to see what happened. All right. Okay. So now this is a citrus cycle, and if you want to guess what will be my requirement for you to know, let me just highlight it. Obviously, you're going to need to know this, right? The acetyl-CoA goes into this. And what am I really care about? Well, what's coming out? CO2, NADH, and NADH, and CO2. So acetyl-CoA is a 2-carbon molecule. By this stage, the 2 carbons are already completely burned into CO2, right? And then, but there's still more energy to be gained from this flow. Now, so you have succinate CoA and succinate, and then you have further burn into releases. These are all high energy molecule, right? So now, and you continue to release electrons, electron released, and by the time here, it's 4-carbon molecule, oxaloacetate. In this process, there's no more energy to be gained, and then this is ready to receive another acetyl-CoA to start the process. And so this is a cycle. What is also important, I wanted to point out, in addition to what comes out, all these are important later on, it is a very important concept that a bacteria, I don't have a bottle here, I have a cup. Imagine this is my bacteria, and there is no structure inside. Everything is floating. So think of these enzymes. They don't stay next to each other. They're in a liquid, okay? And they're floating. And so you have to understand these enzymes are finding the substrate, making them happening. And so this is a process, but it's not a perfect assembly line process because they're not physically tethered together. Okay, this becomes important later on because all these intermediates can be used for something else. Okay, because these things are floating. Okay, that's important concept as well. And so we talked about the NADH before, the electron carrier. And then in addition to that, you have FM, that FADH that can carry electrons as well. So the difference is here. The difference is here. Okay. Okay, so we just went through the citric acid cycle. So now our focus is on this. You generate CO2, NADH, and FADH. I'll leave you to calculate how many later on we'll actually calculate. But it is important to know how many. Okay, so now we're going to look at the electron transport chain. The electron transport chain is actually a misnomer. Yes, it gets to transport electron, but biologically, that's not the entire purpose. The reason you want to have this is not you want to transfer electron, but let's take a look. So we take NADH as an example. So NADH carries 2 protons and then 2 electrons. And so the 2 electrons then gets over here, and then the protons then comes out. All right, look at this arrow. So the 2 protons from NADH actually goes out. This is the outside of cell, this is the inside. So this is outside, out, and this is in, okay? Yeah, it's here. And then the electron continue to flow. Okay, and in the process, the proton gets pumped out. And so from 1 NADH, you actually get to pump out 5 proton. And the electrons doesn't disappear, does not disappear, right? It has to go somewhere. Well, where does it go? Well, the electron goes here, oxygen. Okay, so the oxygen, so the electron gave it to proton and oxygen, and eventually, and so in this process, 1 proton pumps out, 2 protons stay, and to couple with the oxygen ion to make water. Okay, so now you have some inklings on why we have to breathe, right? Can we breathe? Oh, well, we have to breathe if you and the reason we breathe and we need oxygen is our mitochondria, which is carry out this burning of the glucose, requires the electron, requires oxygen as an electron carrier, a receptor. Okay, so now you understand this is the electron transport chain. But the function of electron transport chain biologically is actually to generate proton gradient. Okay. Now it is very important, as I emphasize, that this class focuses on what goes in, what comes out. Right? Electron goes in from NADH. Where does it come out? Well, it comes out here, and to whom? To the oxygen. Because this is important later on. What if bacteria, what if you don't have oxygen? If we don't have oxygen, we're dead, right? But when bacteria, later on I'll explain, some bacteria actually able to use other molecule as electron receptor for the instead of oxygen. Okay, so now we're at stage of talking about electron transport chain, and you understand the electron goes from NADH or FADH goes to oxygen, and in the process they generate proton gradient. Why do we need proton gradient? Well, this is what's going on. It is important that this is the outside, this is the inside. I wish them flipped because all the other format is like that. But anyhow, here is 3 protons is needed to go cross the membrane through the ATPase to generate 1 ATP. Okay, and this is important for your calculation. So if you so this slide summarizes all the energy generates from glucose to the complete burning of CO2. Okay, and generates 38 ATP. Okay, now if there's any other slides that is very important, it is this slides. You should be able to, I don't need you to memorize the intermediates, but you do need to understand how each of these energy molecules are generated. So I would ask you to get a piece of blank paper and then be able to write this out, this table out, do the calculation. Okay. And so that will help you understand where each of the energy will come from. And so now we'll go back. We have I thought I had a suspension, but Sean actually gave you a hint on why do we have to choose. So now you understand there's actually a choice. You have 2 ATP versus 38 ATP. Normally, you would have to go with the one that gives you more ATP, right? But the question is, why would 2 ATP even a choice? So is it all clear to everyone? What determines when E. coli decide whether it should go with the Tca cycle or glycolysis cycle? Anyone want to give it a try? And I get a chance to sip water. Exactly. Very nice. Exactly. Whether you have oxygen or not, right? I wish we have that choice, right? If we don't have oxygen, we just go ferment. But no, we don't. if we don't have oxygen, we are dead, right? But not E. coli. Okay. So the energy generation is very important. We just talked about how use proton gradient to generate energy, but there are other ways of generating ATP, but not very much. And so first of all, you need to understand the energy, where the energy, the most of the energy coming from. Okay, so these are the high energy bonds. And so this is glucose, I'm sorry, ATP, but these are other high energy molecules. And so the way you think about this is the energy that's stored here. So this table gives you the relative energy that's stored in the molecule. So the point of this table is to tell you, allow you to look it up and say, which one is the higher energy molecule? Because only the high energy molecule can give energy to the other to ATP. So according to this table, there are 3 molecules. The first one is phosphoenolpyruvate. And that's a molecule that can generate energy, give energy to ATP because it has higher energy than ATP. So if the phosphoenolpyruvate runs into ADP, it will change ADP into ATP. So that's a process called the the substrate level phosphorylation. And if I may go back to so this molecule here is, sorry, the phosphoenolpyruvate. Okay, so you see this molecule actually gives up the energy to ADP to make ATP, and it becomes pyruvate. Right? Once you give up all the energy, the phosphate bond, and that's the high energy molecule. All right. So I just explained where the phosphoenolpyruvate is and how it's give the energy. So summary, you have the proton gradient to generate ATP, but you also have the what's called the substrate level phosphorylation, and that means the high energy molecule can give the energy to ADP, well, to make ATP, all right? And that's different from the oxidative phosphorylation. So now you learn another name. Oxidative just means you oxidize glucose to generate proton gradient and make energy that way. Okay, so now we know glucose can be used to generate the ATP. But remember earlier I said I used my cup as an analogy of bacteria, a solid phase bacteria, and everything floats, right? And it depends on who gets the substrate or intermediate first. So the glucose is floating around. Yes, it can be used by enzymes in the Myrov pathway to make pyruvate and generate energy, but it can also be used by other enzymes to make DNA and RNA, right? You say, wait a minute. Well, we do need DNA. Well, bacteria, when they grow, they need DNA and RNA, right? How do they do that? Well, they use glucose. And so they go from glucose to ribulose phosphate, and they burn off a CO2. And this is a energy favorite reaction, and then go from there to ribose, and then to ribonucleotide. And the ribonucleotide gets synthesized to RNA. And then the ribonucleotide can also be further oxidized to make deoxy ribonucleotide, right? Deoxy. And so that's why the DNA is. And so now you have RNA, you have DNA. But that's the sugar part that's important. Okay, that's how you make DNA. So DNA and RNA. But we also know that for cell to grow, we need huge numbers of amino acids. How do you get the amino acids? Well, so here's the how you get it. So remember citric acid, right? So the Tca cycle. The Tca cycle, remember, goes from 6 carbon to 5 carbon to 4 carbon, right? So now you have intermediates. You have this is a 5 carbon molecule, and this is 4 carbon molecule. And the amino acids, these are different. These are the 5 carbon amino acids. These are the 4 carbon amino acids. What about the 3 carbon? Well, you have these are the 3 carbon. Okay, so these are the 3 carbon molecules, and then you have even 2 carbon or other molecules, and then the ribose can actually be used directly to make histidine. So this is um, a really brief explanation of the carbon skeleton of amino acids. So how do you make amino acids, right? So basically, you take intermediates from whatever is floating, right? Remember, we said the Tca cycle is the intermediates floating in water, and the enzymes can get to it to either burn energy to complete the Tca cycle, or you have another enzyme can simply add an amine group or a sidechain to modify to make amino acids. Okay, so it depends on which enzyme get to it first. You get to make different things. Okay, so that's how you get amino acids. So now you have amino acids made, nucleotides made, and then you can start thinking about the larger process, making macromolecules. So macromolecules like DNA, right, and proteins, which we'll come back to talk about, because proteins are essentially polymers of amino acids, and DNA is polymers of nucleotides. Okay, so up to this point, what we have just talked about is metabolism. We talked about the metabolism, and we separate them into catabolism. Mostly, we spend time talking about catabolism because how energy is generated, the substrate is generated, and we touched on the anabolism, which is taking the small unit molecule and making larger molecule, which I'll come back to for detailed analysis. And so, so these are the topics we talked about. The glucose focus on generating energy, and the glucose for making the substrate. And we spend most of time talking about how glucose can generate energy, 2 ATP versus 38 ATP, and I highlighted that you want to figure out how the 38 ATP will be generated, and you will, by me emphasizing so much, you know there will be a question on the 38 ATP. Okay, so the next is talking about the growth. All right, so you get energy, and obviously the bacteria need to grow. As I alluded to in previous lectures, bacteria have no noble goals. All they want is grow. Grow, 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 grow. Okay? There's nothing else. All they care about. They just want to grow. Doesn't matter how they want to steal, compete, and grab energy. All they want is grow. Okay, so now we're going to take a look at how they actually grow, and then we'll talk about how we can measure their growth, and then we'll talk about what are the effects, what are the environmental factors that are affecting the growth. So let's talk about the growth first. How do bacteria grow? And the growth is actually pretty simple. In the simplest term, it just goes from 1 to 2. Okay. However, that's what most general public will know, or when you get, before you get into the class, you already know the bacteria goes from 1 to 2. However, in this class, what you need to understand that go from 1 to 2 is actually a very complex process, right? And you need to duplicate everything. Well, first of all, you need to duplicate DNA. And as you're duplicating the DNA, the two DNA molecule needs to be physically separated into two daughter cells, right? If you, I'm sure you have images of your chromosome being poured into spindle structure into two cells, right? Two cells in the eukaryote cell or our cells, right? But for bacteria, it is very important that you notice that this black dots, what does it do? It's a protein complex. It attaches, it tells you that the bacteria DNA, chromosome DNA, is attached to the cell membrane. And that is because the cells are being pulled apart. That's the mechanism. Mechanically, that's how DNA gets pulled apart into two daughter cells to make sure each daughter gets a copy, because if that doesn't happen, the bacteria is dead. Okay, so it's not as simple as goes from 1 to 2. Just like take one cell, you chopped into 2. No, you duplicate, and okay. So now you're talking about this is from 1 to 2 is a 1 generation. Now, the generation time is a measurement of how fast a bacteria grow. So what is our favorite microorganism that we've been talking about? That would be E. coli, right? Yes, E. coli. You do know how to spell Escherichia coli, right? E represents something, right? Okay, just want to make sure. Now, E. coli, we already know a lot about E. coli. We know the size, right? The shape. Now we need to know how fast it grows under optimal condition. So what is the optimal condition? What is the optimal temperature for E. coli? Anyone want to guess? 26? No. Think about E. coli's optimal temperature. Now, you think, how do you address this question? Well, you think about microbes. You think, what is their optimal condition? Well, that must be at their natural environment, because all microbes adapt to their own environment. So here you have Maria says 37. Why? Any? Maria, you want to say why? It's 37? Or anyone else wanted to chime in? You can't go guess. You have to have a reason. Ah, excellent. Human body temperature, yes. E. coli sits in our intestinal tract, and that's its natural environment, right? And so, yes, 37 it is. And so how fast? Well, 30 minutes. At 37 with all the nutrient, E. coli, well, the generation time for E. coli is 30 minutes. 30 minutes at 37 degrees with all the necessary nutrient. Okay, so that's how fast they duplicate. Now, the bacteria, the division is actually pretty complex. And so the E. coli divides in the middle, right? But how does it know where's the middle? It doesn't have a brain, just so, right? And so as it turned out, it has a protein called the Tca protein that forms a ring around it, around the middle. Okay, you have a professor in City College actually studies this one particular protein. She's my best friend. So if you label the Tca here, you can actually see the protein is in the middle of the cell. Right? Now just test you on your microscope skill. What microscope is used to generate this image or this image? Anyone? What kind of microscope? Well, you have light microscope, electron microscope, right? And if you go further, light microscope can be further modified into what? The phase contrast, dark field, fluorescence, and confocal, right? And this probably is a fluorescence microscope or even a confocal, okay? And so the main point here is that the site of cell division is actually marked by a particular protein. And then you can always ask the question, okay, how does this Fitz protein know where to bind? Well, the protein flows around, right? Well, why does this protein doesn't go into the pole, doesn't go here and ends up here? Well, there's other protein guiding it through. Well, how does the other protein guide through? Well, maybe there's another protein guiding through. Now, so as it turned out, the more we learn about microbes, they learn that everything, as I said earlier, that everything in a microbes is hardwired. It just goes from one particular protein to the next. Everything is sequential. Okay? All right. So then take a look at how cell divide, and this is actually very important. So Fitzy protein ring sits in the middle, and that's where the cell division starts. And the green area is where the new cell is, the green area, new cell wall. Okay, so these are the new cell wall, and this is the growing area. This is the old cell wall. And this is how they actually look like under the SEM, scanning electron microscope. Okay, this is a gram-positive streptococcus cell. That's how it grows. Okay, this is very important. So this is the growing area, and this is new area that's growing. Let's take a look. And so we're going to need to look at how they actually grow. And last time we talked about the cell wall structure, and we talked about a peptidoglycan. And so for peptidoglycan cell wall to grow, right? And if you go back here, conceptually in the middle here, this is the growing point, right? Or here. That's where the new cell wall is expanding. And so in order to expand, then you have to separate the original cell wall or peptidoglycan, right? You have to have a gap in the middle, because you have to have a new one added to the middle, right? So this needs to be added. So then the cell wall can expand, and that's how they grow. So the unit is being carried, the new peptidoglycan unit is carried over and then placed in here, right? Placed here. And another way, and then once there, they have to form the new, so cross-link has to be formed. Okay, this is how they form the the sidechain cross-linking, right? Last time we said how it's added. And so this is 2-alanine at the end, and this is very important. And this is recognized by enzymes that that's carry out the process. As it turned out that penicillin is a molecule, you recognize penicillin, right? It's a antibiotic. What does it do? Well, it's an analog for this substrate. So when you have penicillin around, and what will happen is that it will bind to the enzyme that does the transpeptidation, this crossing. And then the penicillin can actually block this reaction. And what happens if it blocks? Well, there is no linkage. Okay, there's no linkage here. Now I'm saying, why is that important? Okay, well, it is important because if we come back here, when you have penicillin, what happens is that the new cell wall, this part, the peptidoglycan is not cross-linked. Okay, only this part, this is the old cell wall. Yeah, it is cross-linked, but this new cell wall here is not cross-linked, meaning the cell wall is very weak. Okay, the analogy is that you build this. Remember, we said the analogy of making a fence, right? You put the sticks down, you use hard metal wire to link them together so that they are strong. So in this case, the metal wire was not there, okay, when you have penicillin. So then when bacteria grow, well, they're going to explode, right? And so when they grow so much, and they just explode, okay? And this opens up, the cell wall no longer holds them and blows up. Okay, one way to imagine this is that if you have a balloon, put them in a cardboard box, right? And then you seal the cardboard box provides the balloon with a structure so that you can pump a lot more air. But if you have a drop of water, or if you soak the cardboard box on one spot, right? Now, if you keep pumping the air, that wet spot the air is gonna come out, or the balloon is gonna explode from there, right? Because the cardboard box, when it's wet, it no longer holds its structure, and then that explodes. Now, when bacteria blows a hole, well, they're dead, right? Okay, so that's that's what we talked about, the peptidoglycan synthesis and cell division, and then we highlighted penicillin, why that's important. So now we're thinking about growth. We said the bacteria growth generation time is at 30 minutes for E. coli, right? And meaning there are other bacteria can grow faster or slower, right? So we need to look at the growth rate. How do you actually track them? So how do you measure bacterial growth? Well, you measure by how fast they generating cells, right? And you count the numbers of cells. Well, and if you know the numbers, and then you can plot them. So when you plot them, there are two different ways of plotting. And one is if you do the arithmetic plotting, okay? So this is the number of arithmetic scale, okay? From 100, 500 to 1000. So that's the green line, okay? So the green goes here, and if you plot that number, that's the curve you will see. However, if you convert that number into a log scale, and then you plot the number, and then you actually get a straight line. And so why is that important? Well, the slope of this line will visually tell you how fast the bacteria is growing. Because if you have a bacteria and the number here, and if you have another bacteria that gives you 10 cells for the same period of time, then the curve will look like this, right? So if you compare the red curve versus the blue curve, it's so obvious that blue curve tells you that this blue bacteria is growing very slow. Okay, so from that point of view, because of that, the microbiologists and going forward, all the growth curve is plotted using log scale. Okay, so everything is log scale. And so if you okay, so that's the growth rate and how we track them. Then how do you study the bacterial growth cycle? When we think about bacterial growth, we typically don't say individual, we say population, because the bacteria goes from 1 to 2 cells, and that's in 30 minutes, right? But if you have a population bacteria, and that actually can increase a lot more, and that's the part that we care about, how bacterial population how fast they can expand their population. So then we are interested in that population. So if you put a bacteria into a test tube or a flask with media, and then you let them grow, and that's called the batch culture. Okay, so the batch culture refers to a population bacteria in a defined amount of media, like one time. This is different. This is batch culture is in contrast to continuous culture. There's another culture called continuous culture, where the media is continuously being supplied with nutrient. All right. So so this is when we look at the batch culture, when you put the bacteria into a flask with media, and the bacteria doesn't immediately grow. Okay? And if you follow the green line, which I'll explain a bit later, if you follow the green line, what would you see is that the bacteria will stay flat for a while. Well, why? Well, because it has to adapt into the environment, and it does take time to make up new things, right? And remember, 30 minutes for E. coli to start growing, and once they get to grow, once they adapt into this new growth environment, then they would go go into an exponential growth phase, where would they go by 30 minutes per generation, 1 to 2, And so this area, this is more or less a straight line. And this slope determines what tells us, visualizes how fast the bacteria is growing. And now when they get to stationary phase, meaning the nutrient gets less and less, meaning the glucose, if you put a bacteria in the glucose, and at some point the glucose will reduce, right, and use up, and then they go into stationary phase. All right, they go very slow, the population doesn't increase, and after the stationary phase, the bacteria actually go into death phase. Well, why? Well, because they actually will die. They run out of nutrient, they will die. That's for E. coli. And if we use bacillus, earlier on last week we talked about the endospore, and that's when they're producing endospore. Okay, so the green line is where we track the bacteria using what's called the optical density, the turbidity. So that comes into a question about how do you actually track and count the numbers of bacteria. I said, well, the number doesn't increase, but here we use an optical density to track. But there are other ways of tracking bacteria. Okay, next I will tell you, I would like to discuss how do we measure bacterial growth. And so one way is to simply count the numbers of bacteria in there. And so this is a counting cell. It's a special slide. And then, so what happened here is they specially make the slide look like this. So this is, I'm sorry, a terrible drawing. And so these are the glass. Okay, so I meant to say glass, glass, glass. And then you have another glass on top, cover slip. Okay. So now what I'm trying to show you is, let me see if I can change the color. Notice this particular area, the distance, we just manufactured so that this part is lower than the side, and we determine how high. And so now if you put the liquid cell suspension in here, and then you count the numbers of then you look down from the top and you know the area. So if you know the area, you know the height, what do you get? You can calculate the volume. And so this grid tells you, looking down, you can actually see how many cells in there, and you can convert that to the volume, and that's how you generate the cell concentration. Okay, so the advantage of this using counts, and sometimes people still use more common people in a lab using a cell counter to count, say, red blood cells, and that's what mostly commonly seen. For bacteria, we don't use that as often, but partially for microbes, for direct counting, you have the advantage of very fast. You know immediately how many cells per ml. The downside is that if this is bacteria, then you may not be able to tell how many bacteria is alive or dead. Right? That's the downside or limitation of direct counting. For bacteria, we're mostly interested in how many living bacteria are there in the solution. So what we do, then you spread them onto a plate. when individual bacteria lands in a spot, and they will grow into a colony, right? So that one colony spot represents one original bacteria at that particular spot. Because on the solid surface, the bacteria won't move, and they just become a pile of bacteria, okay? And so then the other way is to mix the bacteria suspension in the in agar. And so now so this is a pour plate, and this is spread plate. And so pour plate, and that will have bacteria embedded in the middle of agar. And the colonies, because they are constrained by the agar in the middle, they actually will be physically constrained so that they can't get any bigger. The cell may be dense, but they actually get limited in size, versus the colonies on the surface gets bigger. Okay. Now, each colony takes up a particular size, right? Because otherwise it wouldn't be visible. Well, because if you have too many colonies, they eventually fuse together, and then it's difficult to count. So you have to do dilutions. You have to dilute them. But if you don't know what's the original cell concentration, you just automatically do 10 to the 6 stepwise dilution. And then you can count, you put 1 mL onto each of the plate, and then you can count how many bacteria on each of the plate, and then you can then count back each the colonies, and then you calculate back to see based on the dilutions, and then you calculate back what are the actual cell concentrations in there. All right. So that's a spread plate, and that the dilutions makes it possible to do spread plate print counting, spreading counting. Okay. And so those are the direct measurement, but you can actually have indirect estimate. That's the optical density. Okay, so the optical density essentially just like if there's cell suspension, you can even hold up to the light and say, it's cloudy, right? Cloudy is because it's blocking the light. So that then we use a spectral photometer. I'm sure you've had this in different different labs. You have optical density, you can measure them. And so the optical density is usually a measurement of how dense a bacterial culture is. Typically, the bacteria uses OD 600. OD 600. And that 600 represents the nanometer of the light that's used. So we use a filter to select a particular wavelength of light to measure cell density. And so for bacterial cell, we typically use the 600 nanometers wavelength to measure colony density. And so if you were to use optical density, then you can see one bacteria A and B, and you can see that A is growing faster and B is growing slower. And the advantage of optical density, while it's not precise, and is it's fast. The downside is, again, you run into a problem that the dead bacteria maintains the optical density, because the dead bacteria doesn't lyse immediately. So I'm back into this slides. And so now you understand the optical density is measuring tubility, right? Total numbers of cells, living and dead. The viable count is counting only the numbers of cells that are alive. It's this tubility count is actually, the difference is actually much more dramatic than this. So this, it should be, say, you see green, the green light, it should be actually pretty flat for a long time. And then the red, because they're dying, and the red line should go down a lot more dramatic, and because they die. Eventually, the red line actually reached to almost to no living cells, if you starve them long enough. Okay, so now you understand the at the beginning the viable count stays flat and goes up, and then when you get to stationary phase, the number stays flat. But the difference is at the death phase. The tubility count the total living and dead cell. The viable count counts the living cells only, and the curve will be different. Okay. So those are the two ways of measuring bacterial growth. Okay, so we now talked about the bacterial growth and how cell growing, how cell divide, and we focus on peptidoglycan synthesis and the effect of penicillin. And then we talked about the growth rate and growth cycle and how do we actually measure them. Okay, so the next I wanted to briefly discuss, it's pretty straightforward in terms of what are the factors that can affect the bacterial growth. All right, and the first is the temperature. As it turned out, we know on Earth there are hot spots and cold spots, right? So there are places are very, very cold, and there are places very, very hot, right? And where's the E. coli? Well, E. coli is here. Let's see, I want the blue one. And so the E. coli is here, right? But there are hotter places, like hot spring, and very, very hot spring. And then there are places that's so hot. Where would you find 106 degrees Celsius? Right? We're talking about Celsius. Where would you find 106? Volcanoes. Yeah, volcanoes is hotter than that, probably thousands of degrees if you think about Hawaii's and lavas and coming out, right? Anyway, well, nothing will grow. Under the water vent. Excellent. Okay, but why, this may be asking your understanding of physics. Why does the temperature underwater vent can get to 106 degrees? That gives me a second to drink some water.
Is it
Excellent.
because of the lava coming up? Like, not the lava, but like the earth core being the thinnest there?
No. It's the reason the temperature the water boils at 100 degrees, right? That's on the sea level. But if the water goes to under the sea, you know, hundreds of meters down, the pressure is enormous, right? Because of the water column. And so when you have high pressure, the water can actually, the boiling temperature can be actually higher. And so that's why the temperature can get to more than 100 degrees in undersea water vent. Okay, so now there are different temperatures. That's good. And there are different microbes adapt to different environment. It is very important that you understand the adapting to that particular environment. Why? Because if you change the temperature for one particular microbes, and they will not be able to grow too far away from the optimal temperature. Okay, so if conceptually this fits everything, so this curve fits everything, and there's an optimal growth temperature and there's a maximum. When you get to maximum temperature, the protein gets denatured, right? Your egg turns into white, the membrane and they just lies, right? But as from, so that's on one direction of too high, but what happens to too low? At the end, when it gets to actually too low, the membrane gets gelled, forms a gel. And transport process became so slow. And this gelling part is very important. Why? Well, because remember, enzymatic reaction depends on enzyme finding the substrate and then carries out the reaction, right? And the enzyme needs an ATP. Well, now, if it's in a gel, that means everything is in suspense, no relative movement. The enzyme stays alone, wouldn't be able to find the ATP or the substrate. So there is no growth, right? So, but in between, the enzymatic reaction will increase, right? And as when it gets to the optimal temperature. And so this applies to everything. So if you put E. coli as an example, when you get to 45 or 50 degree, and E. coli's protein will simply denature. But if you put E. coli, on the other hand, into a fridge at 4 degree, and because its optimal temperature is at 37, 39, the E. coli cells, cytoplasm becomes a gel. Well, then obviously it won't be able to grow. So now you understand E. coli, if you put E. coli into a refrigerator, what will happen is it just not grow, it just stays. Okay? So, so wouldn't they evolve? Oh, so the question is, if you put E. coli into a very, very high temperature, can they evolve? Okay, yes and no. You have to understand why other microbes can grow. So if you want to put E. coli to grow at 60 degrees, what you will need to do? Well, the protein denatures. Why do they denature? Well, because the protein used to rely on hydrogen bonds to stay together as a structure. But now, when the temperature goes too high, the hydrogen bond won't be able to strong enough to keep them together. So you need disulfide bonds. So for proteins at this temperature, the bacillus, they will have the temperature. The protein will have the temperature. I'm sorry. The proteins will have a lot more disulfide bonds so that the protein can stay stable at 60 degrees. Now you can have even higher when it gets to higher temperature. Well, you need more disulfide bonds and more disulfide bonds, right? Now, to get a protein to increase a disulfide bond, that's not a trivial thing. And it takes probably millions of years of evolution to get to that stage. So a gradual continuous increase of temperature over a long period of time actually will select the changes. Okay, so if you put E. coli into a different temperature, they just died. And if you put this thermal cellular bacteria into a regular temperature, it wouldn't live as well. It would just not. It would just stop. Okay, so if you put a hot bacteria lives in hot spring, like here, into a regular water, 26 degree, and what would they do? They wouldn't be able to grow. Okay, here are some nice pictures of spring boiling water, and you can actually see bacteria in there. And this is cold, very, very cold in snow. And they can grow. Now, I said you put E. coli into a fridge, they won't be able to grow. But what happens if you put E. coli into a freezer? Will they be able to survive a freezer? Are they preserved? What happens when if you put water into a freezer? What happens to water? The water forms, yeah, it freezes. And then that's not what I'm looking for. So think about snowflakes is what? Crystallize. Yes, that's what we're looking for. When water starts freeze on ice, if you think about snowflakes, right? They form a needle-like structure. Think about those needles crossing the bacterial cell membrane, and those can actually punch holes in cell membrane, right? So they form holes in the cell membrane, and then the bacteria, when they thaw, now the bacteria is all holes, and then they're dead. So there are ways of avoiding this. So glycerol is one molecule, and DMSO, the glycerol is more common. So if you mix glycerol with bacteria, and the glycerol actually goes into the bacteria and mix with cytoplasm, and then isolates the molecule into smaller droplets. And when you freeze those bacteria, so when you freeze bacteria in the presence of glycerol, then these the water crystals will not form, and so then the membrane will remain intact. So in practice, what actually we do is that if we want to stop the bacteria, we want to save the bacteria or preserve the bacteria, we put them in 10% glycerol, and then mix with 10% glycerol, and then we put them in minus 80 degrees Celsius freezer, and then we can keep them alive for a long time. Okay. Now there are other factors very important. pH, which is very obvious. The key here is that the bacteria can withstand to 1 pH. So if the bacteria adapting to pH 7, they will be able to grow in pH 6, so this range, up and down 1. And then they can survive up and down 2. So they won't be able to grow between 5 and 6, but they would, stay alive. They can be revived. And then you have osmotic pressure. Okay, osmotic pressure meaning the salt. And so E. coli, for example, is very, very sensitive to salt concentration change. If you increase salt concentration, E. coli die. And Staphylococcus actually doesn't mind go to higher temperature, the salt concentration. And Vibrio fischeri is a seawater microbes, and it can withstand very high concentration of salt, but Halobacterial cellarum is actually a purple bacteria, extreme halophile. It loves the salt concentration. It lives in the salt field. Later on, we'll come back to talk about this. But this is all related to their tolerance is also related to their environment. Think about that. And our salt concentration in our gut hopefully doesn't change dramatically. And that's why E. coli doesn't have the ability to adapt into high salt concentration. But Staphylococcus is a common bacteria that we carry. Where do they live? Anyone? On the skin, in your nose and in your eyes. And on your skin dries up really fast, right? That's where the salt concentration will increase. And forget about if you have to go and run and you sweat and then you sit and then you have a crust of salt on your skin, right? Just to let you visualize how salty your skin could be. Okay, and the Staphylococcus has to withstand those changes, right? And then you have oxygen. Oxygen is important, right? We talked about it determines whether E. coli will go fermentation or like oxidative pathway, right? Tca cycle. And as it turned out, different microbes live in different environment, adapt into their own environment, right? Because you can have bacteria live on with lots of oxygen, but if bacteria live in the mud, there's no oxygen there, all right? And so based on the oxygen requirement, the bacteria can be separated into at least 5 different groups. And so this is an experiment that you will need to learn a little bit more about this. So let me explain. So these tubes contains agar. So these are solid, not liquid. These are solid. And what they did here is they mixed bacteria into solid agar and then poured into this tube and then put a nutrient, a layer to seal it on the top. And then they let them grow. And so after the incubation, you can see colonies. So these black spots represent colonies, okay? These black spots, not individual cells, they are colonies. You have individual cells here too. They just didn't grow. So in tube A, the bacteria, when you mix them together, only grow on the surface. And this bacteria only grows at the bottom. And this bacteria grows throughout. And so the reason they're different is because the oxygen. The oxygen is available on the top. It can diffuse into the bottom. And so this bacteria then, because it only grows in the presence of oxygen, so it's an aerobe. And this bacteria only grows in the absence of oxygen, is anaerobe. And this bacteria, then look at this bacteria first, and this one. And they grow not on the top, not at the bottom, but in the middle. So this is called the microaerophilic. Needs a little bit of oxygen. And then if you look at the C and the E, if you notice the difference, the E has the cell colony density the similar all throughout, versus here you have more colony on the top. That should tell you that this bacteria can grow in the presence of oxygen and in the absence of oxygen. So this is a facultative anaerobe. And this bacteria can grow both. But here they use oxygen. Here it doesn't use oxygen. It aerotolerant. So this bacteria tolerates the oxygen, but it doesn't use oxygen to grow. Okay, so these are the 5 different groups, major groups of microbes based on oxygen, the needs of the oxygen. Now, in labs, you can create anaerobic environment by putting the petri dish in a sealed chamber, and we actually have them in the lab. And then you put gas pack. It's a chemical that consumes oxygen. Chemically, use up all the oxygen, and then there's no oxygen here, and that creates an anaerobic chamber, and so the bacteria can grow. However, when you have oxygen around, or even when you do the oxidative process, you can have reactive oxygen that's generated. Okay. So then the oxygen needs to be removed. And so these are 4 different steps of oxygen reduction to go from oxygen to, because these reactive oxygen can actually react to DNA or other proteins in the cell and actually kill them, right? You recognize this particular molecule? Hydrogen peroxide. Yes, that's the molecule. Oh, that's the thing that you get from CVS or Walgreens or Rite Aid. In case if you have a cut, you use hydrogen peroxide to do what? To sterilize the wound surface, right? It's painful, I know. All right, but that's hydrogen peroxide, and you can add it to the bacteria, and that could kill the bacteria. Now, bacteria are very smart. They actually have ways of deal with these reactive oxygen. Catalase or peroxidase are the enzyme. The bacteria can produce enzyme that actually turns hydrogen peroxide into water. Well, this is a defense mechanism so that those aeron-tolerant microbes, right, or aerobic bacteria, they can actually remove the toxic hydrogen peroxide that's generated during the metabolic process. So these are examples of the enzyme that can be used that some microbes will have. One of the ways you can test a bacteria whether they are producing catalase is to simply put a hydrogen peroxide onto the bacterial colony. And you can actually physically see the bubbling. It's a really interesting experiment. Okay. Maybe in the future when you come back in, no, actually you guys will very lucky can actually do it in the lab. We'll actually show you. So these are the 4 main factors that can affect the microbial growth. Okay, and so I'm gonna stop the lecture here.